It starts with almost nothing, just one cell. A fertilized egg. It's a tiny sphere, smaller than the period at the end of this sentence. It holds no thoughts. It has no memories. It feels nothing. It is, for all purposes, just a starting point. And yet, locked inside this single, simple cell is the complete set of instructions to build the most complex object in the known universe. A human brain. How does this happen? How do you get from that one single point to 100 billion neurons connected in a network of 100 trillion links? How do you build a machine that can write poetry, calculate the distance to the stars, feel love, and ask a question like this one? It's not magic, it's a process, it's a biological construction project of unimaginable scale, guided by a few core principles. It's a story of multiplication, specialization, migration, connection, and finally, sculpting. And it all begins with the instructions. Every cell in your body, including that first one, contains your DNA. People often call DNA a blueprint, but that's not quite right. A blueprint is a fixed plan. It shows exactly where every wall, every wire, every pipe goes. If the blueprint shows a wire going from point A to point B, that's where the builder puts it. The brain isn't built like that. A better way to think of DNA is as a recipe book. Or maybe an instruction manual for a set of tools. It doesn't say place neuron number 5,342,111 at these exact coordinates. It says things like, when you sense chemical to 10th, turn on gene Y, and gene Y might be the recipe for a protein that tells the cell to move, or to become a certain type of neuron, or to connect with a neighbor. The instructions are conditional. They are rules of engagement, not a static map. So, the process begins. That first cell divides into 2, then 4, then 8, 16. This is proliferation. It's the first and simplest step making the raw materials. The cells are just copying themselves again and again. In the early days of the embryo, this growth is explosive. A single cell becomes a small ball of identical cells. They are all generic, all potential. They all have the same complete DNA recipe book inside them, but a brain made of identical cells is useless. You need different kinds of cells. You need neurons to carry signals. You need glial cells to support and protect the neurons, to clean up waste, and to help signals travel faster. So the next step has to happen. It's called differentiation. This is where the recipe book analogy really works. Imagine you have 100 cooks in a kitchen. They all have the exact same massive cookbook. If they all cook the same recipe, you just get a mountain of one dish. To make a full meal, you need a head chef to go around and tell each cook which recipe to use. You make the sauce. You prepare the vegetables. You bake the bread. In the developing embryo, there is no head chef. Instead, the cells get their instructions from their environment and their neighbors. A cell's location is everything. Depending on where it is in that growing ball of cells, it will be exposed to different chemical signals called morphogens. Think of these chemicals as spreading out from a source, getting weaker the farther they go. So a cell might find itself in a place with a high concentration of chemical A and a low concentration of chemical B. This unique chemical signature acts like a trigger. It flips a switch inside the cell. It tells the cell, open the cookbook to the chapter on becoming a motor neuron. A different cell, in a different location with a different chemical mix, gets a different instruction. You open the chapter on becoming a glial cell. This process is called gene expression. Even though every cell has all the genes, only some of them are turned on or expressed. The rest are silent. This is how you get hundreds of different types of cells in the body from a single starting cell. The plumber and the electrician both have the same full set of construction manuals, but the plumber only reads the plumbing chapters, and the electrician only reads the electrical chapters. Their job is defined by the instructions they actually use. So now we have cells multiplying and specializing. We're making the bricks and the wires and the pipes, but they are still just in a pile. The next challenge is to give the brain its basic shape. A brain isn't a random soup of neurons. It has a front and a back, a top and a bottom. It has a left and a right. It has distinct regions, a forebrain for thinking, a midbrain for basic functions, a hindbrain for coordination. This large-scale organization, called patterning, also uses those chemical gradients. Early in development, the embryo establishes major axes. A source at one end releases a front signal. A source at the other end releases a back signal. Another system sets up a top and bottom axis. Now, every cell in the developing nervous system has a kind of address. It knows if it's in the front top part of the brain or the back bottom part. This address tells the cell what kind of neighborhood it's in and what its general job will be. Cells in the front part are now fated to become part of the cerebral cortex. Cells in the back will form the cerebellum. The grand overarching structure of the brain is being sketched out not by drawing lines, but by establishing invisible fields of chemical information. It's like setting up a grid system over an empty plot of land. You're not building the houses yet, you're just defining where the streets and zones will be. Once the zones are defined and the specialized cells are being made, something truly remarkable happens. The neurons begin a journey. Most neurons are not born in the place where they will spend the rest of their lives. They are born in special neuron factories deep inside the developing brain near the fluid-filled ventricles. Then they have to travel. This is migration. Imagine building a skyscraper. You don't make the bricks on the 50th floor. You make them on the ground and then hoist them up. The developing brain has its own hoisting system. A special type of glial cell, called a radial glia, stretches a long fiber from the inner part of the brain all the way to the outer surface, like a rope or a scaffold. A newborn neuron, a tiny, immature cell, latches onto one of these fibers and begins to climb. It pulls itself along, inching its way through a dense, crowded forest of other cells. It's a perilous journey that can take days. The neuron has to hold on tight, following its guide rope to its predetermined destination. The brain is built from the inside out. The first wave of neurons climbs the ropes and forms the deepest layer. Then a new wave of neurons is born, and they have to climb past that first layer to form a new layer on top. This happens again and again, building up the 6 layers of the cerebral cortex like layers of an onion. If a neuron gets lost, or falls off its rope, or stops too soon, it can lead to developmental problems. The precision of this migration is astonishing. Millions of cells, all crawling to their correct layer in the correct region of the brain, all at the right time. So, now we have the right kinds of cells in the right general places, arranged in the right layers. The city has been zoned, and the workers have arrived at their job sites. But a city isn't a city until the roads and power lines and communication cables are all connected. The next step is the most intricate and mind-boggling of all wiring the brain. Each neuron has to send out a long, thin wire called an axon to connect with other specific neurons. Some of these connections are short, to a cell right next door. Others are incredibly long. An axon might have to travel from one side of the brain to the other, or from the brain all the way down the spinal cord. This is a journey of immense distance, relative to the size of the cell. It's like a person in New York trying to find a specific person in Los Angeles and shake their hand. How does an axon find its way? It doesn't travel randomly. At the very tip of the growing axon is a structure called the growth cone. You can think of it as a microscopic hand or a sniffer dog. It's constantly reaching out, feeling, and smelling its environment. The environment is filled with chemical signposts. Some chemicals attract the growth cone, telling it come this way. These are chemoattractants. Other chemicals repel it, saying not this way, turn back. These are chemrepellents. The path an axon takes is a complex molecular obstacle course. It is guided along a very specific route by a sequence of these attractive and repulsive cues. It might be attracted to a point a little way ahead, so it grows towards it. Once it gets there, that attractive cue might fade, and a new one might appear telling it to turn left. It might encounter a repulsive barrier that forces it to turn right. It navigates through the dense tissue of the brain, a pioneer in a new land, following a chemical map that only it can read. This journey is not a single, straight shot. An axon has to make many decisions along the way. When it reaches a major junction, it has to decide whether to cross to the other side of the brain or stay on the same side. This decision is controlled by specific molecules at that junction. For example, some growth cones have receptors for a stop here signal, while others ignore it and keep going. This is how you get the incredibly precise and complex wiring patterns that define the brain's circuits. And when the axon finally arrives in the correct neighborhood, its job still isn't done. It's not enough to just be near the right cell. It has to find the exact right type of cell and form a connection, a synapse. This is the moment of the handshake. When the growth cone touches a potential partner cell, a molecular conversation takes place. Proteins on the surface of the axon tip interact with proteins on the surface of the target cell. It's like a lock and key. If they match, a signal is sent back to both cells, connection confirmed. Then, the synapse itself is built. It's an incredibly complex piece of molecular machinery. On the axon side, little packets of neurotransmitters, the brain's chemical messengers, are assembled. On the target cell side, receptors designed to catch those specific neurotransmitters are put in place. The two cells don't quite touch. They leave a tiny gap, the synaptic cleft. An entire infrastructure is built around this gap to allow for the reliable transmission of a signal from one cell to the other. This process happens trillions of times. Every neuron forms, on average, thousands of synapses. The result is a wiring diagram of staggering complexity, a network that dwarfs any computer network ever built by humans. But the construction project is still not finished. In fact, what the brain has done so far is create a massive oversupply of everything. The brain makes about twice as many neurons as it will eventually need, and it makes vastly more connections than are useful. The initial wiring is messy and redundant. It's like a sculptor has just quarried a giant block of marble. The basic shape is there, but the fine details are hidden inside. The final step is to carve away the excess. This refining process happens through competition. It's a use it or lose it system. Neurons need to connect to their targets to survive. They receive life-sustaining chemical signals called neurotrophic factors from the cells they connect to. It's like a limited supply of food. If a neuron makes successful connections, it gets fed and survives. If it fails to connect or makes weak connections, it doesn't get enough of these survival factors. When a neuron doesn't get the support it needs, it triggers a program of controlled self-destruction called apoptosis. It neatly dismantles itself and gets cleared away by the brain's cleanup crew. This isn't a mistake, it's a critical part of the plan. It eliminates neurons that are in the wrong place or that failed to wire up correctly. It ensures that the remaining components are the ones that found their proper place in the system. A similar process happens with the synapses. Initially, a target cell might receive connections from many different axons. But over time, only the strongest and most active connections are kept. The ones that are used frequently get strengthened. The ones that are silent or used rarely get weaker and are eventually eliminated. This is synaptic pruning. This sculpting process is heavily influenced by experience. After a baby is born, the brain is still a work in progress. When the baby sees light, the connections in the visual cortex are activated and strengthened. When it hears its mother's voice, the auditory circuits are reinforced. When it moves its limbs, the motor pathways are solidified. The experiences of the outside world are what guide the final stages of the wiring. The brain sets up a rough draft, an explosion of potential connections, and then the environment refines it into a masterpiece. The circuits that are relevant to the world the child is actually living in are the ones that survive. This is why early childhood is such a critical period for development. The brain is literally shaping itself to fit its world. So let's step back and look at the whole story. It starts with one cell and a book of recipes. That cell divides, creating a huge number of generic cells. Chemical signals based on location tell these cells which recipes to read, turning them into specialized neurons and glia. More chemical signals lay out the large-scale map of the brain, giving every cell an address. The newborn neurons then travel, crawling along glial fibers to reach their designated spot in that map. Once there, they send out axons whose growth cones sniff their way through a chemical landscape to find their specific targets. When they find the right partner, they form a synapse, a complex communication junction. This process creates a massive overabundance of cells and connections. Then, a period of pruning begins. Cells that don't connect properly die off. Synapses that aren't used are eliminated. The circuits that are left are the ones that are active, effective, and shaped by experience. From a single cell, an organ is built that can contemplate the universe. It's a process that is both stunningly simple in its principles and bafflingly complex in its execution. The rules are straightforward: multiply, differentiate, migrate, connect, and refine. But applying these rules results in a structure with trillions of moving parts, all working in concert. And what's perhaps most amazing is that there is no master builder, no foreman, no architect standing over the project. It is entirely self-organizing. The rules are embedded in the DNA, and the process unfolds as a consequence of those rules playing out, cell by cell, moment by moment. Each cell is just following its local instructions. It responds to the chemicals in its immediate vicinity. It talks to its neighbors. It doesn't have a concept of the brain it is building. And yet, the collective action of all these individual cells, following their simple rules, results in the emergence of a functioning, thinking brain. We still have so much to learn. We don't know all the chemical cues. We don't understand how trillions of connections self-organize into coherent thoughts and consciousness. We've mapped the major highways, but the individual side streets and the traffic patterns are still largely a mystery. But we know the fundamentals. We know that the journey from a single cell to a complex brain is not a miracle in the sense of breaking the laws of nature. It is a miracle of nature itself. It's a testament to how a simple set of genetic rules, iterated over millions of years of evolution, can give rise to something so profoundly complex that it has the power to look back at itself and wonder how it ever came to be. And that process, that continuous unfolding of life and complexity, is still happening right now, in wombs all over the world, building the minds of the next generation from the humblest of beginnings. A single cell.